Imagine a world where a single injection could erase inherited diseases, restore lost vision, or unlock the body's own immune army to fight cancer. That's not science fiction anymore. It's the breathtaking promise of gene and cell therapies. But there's a problem, hiding in the vaults and labs of the world's most innovative biotech companies, a bottleneck so severe that it could stall the next leap in medical history. If gene therapy is a rocket, there's a crucial part of its engine that remains shockingly fragile. This isn't about talent, funding, or even breakthrough science. It's about the invisible workhorses behind the scenes, the viral vectors that shuttle therapeutic genes into our cells. Right now, the race to supercharge their manufacturing is turning biotech into a battlefield. Let's peel back the curtain on what might be the most urgent, lucrative, and high-stakes arms race in global healthcare. Why are lentiviral vectors, the viral delivery vehicles inspired by HIV, suddenly in the spotlight? Who are the players scrambling to break through the manufacturing ceiling? And what are the risks if they fail? As the costs soar and patient hopes hang in the balance, the scramble to master lentiviral vector production could determine who controls tomorrow's cures and who is left behind. To truly grasp the stakes, let's start with a crash course in the world of viral vectors and the promise and peril of gene therapy. Gene and cell therapies are arguably the most dramatic paradigm shift in medicine since antibiotics. By rewriting faulty genes or engineering cells to fight disease, these treatments are changing what's possible, not just for rare genetic conditions, but for cancer, autoimmune diseases, and even HIV. But unlike conventional drugs, these therapies need to place genetic instructions precisely inside the right human cells. Enter viral vectors, the microscopic vehicles co-opted from nature's best gene delivery machines, viruses themselves. Among these, lentiviral vectors, or LVVs, are the real workhorses. Lentiviruses, most famously HIV, evolved over millions of years to insert their genetic payload into host cells efficiently and precisely. By stripping them of their disease-causing abilities and reprogramming them to carry therapeutic genes, scientists have turned the hunter into the healer. Now, LVVs are becoming the backbone for therapies like CAR-T, where a patient's own immune cells are re-engineered to hunt down cancer, and the only way to reprogram those cells is with a lentiviral vector. The gene therapy industry is exploding. Recent forecasts predict the viral vector manufacturing market alone will skyrocket to over $5 billion by 2028, growing at a staggering 16.8% annually. Behind the scenes, the lion's share of investment is pouring into LVV technology. As the field expands from rare diseases into mainstream cancer treatments, the demand for safe, potent, and scalable viral vectors is cresting to an all-time high. But why lentiviral vectors? In the alphabet soup of gene therapy, adeno-associated viruses, AAVs, have long ruled, especially for treatments delivered directly into the body. Yet, LVVs are now rapidly gaining ground, especially in ex vivo approaches, where cells are taken from the patient, engineered in the lab, and returned. LVVs can carry larger, more complex genetic cargoes and integrate them more stably. This is vital for therapies like CAR-T, where precise, lasting reprogramming of immune cells is a must. Bill Swaney, president of Expression Manufacturing, puts it vividly. Lentiviral vectors are transforming gene therapy by enabling delivery of complex genetic payloads with unprecedented precision. He's not exaggerating. But beneath the surface, the technology has some hard limits. And now, as more therapies aim for approval, these limits are becoming existential threats. What exactly are the core bottlenecks? First, scalability. Manufacturing LVVs remains far more art than science. Current production methods, often adapted from academic labs, are not optimized for industrial scale. Bioplan Associates warns, upstream process development remains the Everest to be conquered for gene therapy manufacturers. Without efficient and scalable upstream processes, the entire field risks stagnation. Second, analytic rigor. Measuring the potency of LVV batches is still a challenge. Accurate titration, quantifying how many infectious particles are present, requires sophisticated analytical methods that remain cumbersome and inconsistent. As Dark Horse Consulting succinctly puts it, a comprehensive and unflinching analytical strategy is the bedrock of product quality and safety in lentiviral vector manufacturing. Wishful thinking is not a substitute for rigorous data. Third, regulatory scrutiny. The US FDA and global agencies are tightening guidelines. As more patients receive gene therapies, the long-term risks, like insertional mutagenesis, where the vector inserts genetic material in the wrong place, demand rigorous follow-up and ironclad risk mitigation. The FDA cautions, proactive and transparent engagement with regulatory agencies is not optional. It is the price of admission for navigating the labyrinthine approval pathways for gene therapies. Ignorance is not bliss, it's a recipe for disaster. And finally, cost. Even as science races ahead, the actual cost of producing effective LVV batches remains eye-watering. Thermo Fisher Scientific, a leader in the space, warns, we must shatter the current lentiviral vector titer ceilings to drastically reduce the cost of goods sold, COGS, for cell and gene therapies. Incremental improvements are no longer sufficient. With so many obstacles, why does the industry persist? Quite simply, whoever cracks the lentiviral vector code could unlock billions in revenue, and more importantly, change the lives of millions. McKinsey & Company offers a stark prediction. The ability to master viral vector gene therapy manufacturing will separate the victors from the vanquished in the coming years. Those who fail to innovate will be left behind. That's why right now, a new wave of strategic alliances and technological moonshots is sweeping the field. One of the most closely watched collaborations is between Wacker Biotech US and Expression Manufacturing. By combining Wacker's PlasmaTech platform, which uses proprietary engineered E. coli to churn out ultra-pure plasmid DNA, the core ingredient for vector assembly, and Expression's lenti platform, designed for safer, more potent viral vector backbone technology, they've promised an end-to-end solution for biopharma clients. Mark Bush, head of business development at Wacker Biotech US, says, we now offer through this partnership the viral vector backbone technology to complete the manufacturing cycle for lentiviral delivery. But even as alliances like these accelerate, the main conflict looms. Is the current technology enough to meet demand? And can the industry overcome the mounting challenges before time runs out? Let's break down the core challenges fueling this high-stakes race. First, the technical ceiling. The manufacturing process for lentiviral vectors is still hobbled by outdated methods. Most production relies on 293 T cells, huge flasks, and batch processing, far from the robust, scalable bioreactor setups that modern biologics use. Yields are limited, contamination risks are real, and the titer ceiling, the maximum concentration of usable vector, remains stubbornly low. Incremental improvements help, but industry voices agree. Radical innovation is needed. Second, the supply chain maze. One of the least discussed but most crucial aspects is the upstream supply of high-quality plasmid DNA, the starting material for making viral vectors. Until recently, most companies relied on third-party suppliers, leading to bottlenecks, delays, and variable quality. Now, insourcing plasmid DNA production is becoming a strategic necessity. Wacker Biotech's investment in a San Diego pDNA Center of Excellence is a sign that the smartest players are securing control over their own raw materials. Third, regulatory and safety hurdles. The push for ever more potent and complex vectors comes with growing concerns about safety. Next-generation LVVs are being engineered with microRNA target sequences to minimize off-target effects and improve safety. But every leap in complexity means more unknowns. And with the FDA demanding not just short-term data but years of patient follow-up, the regulatory burden is immense. Fourth, cost and access. The economics of gene therapy are daunting. While the science dazzles, the price tags, often hundreds of thousands or even millions of dollars per patient, are mostly driven by the costs and inefficiencies in vector manufacturing. Until yields can be massively increased and costs slashed, access to these therapies will be restricted to the wealthiest healthcare systems and a small subset of patients. Finally, the human factor. This is a race, but also a balancing act. Hidden inside the big revenue numbers are real lives, kids with rare immune deficiencies, adults with aggressive cancers, families hoping for a cure. Each delay, each faulty batch, is a setback not just for investors, but for patients on the edge. Tensions are mounting. Established pharmaceutical giants, nimble biotech upstarts, and specialized CDMOs, contract development and manufacturing organizations, are converging on this problem from every angle. Bioprocessing automation, single-use manufacturing systems, new analytical platforms for vector quality, and bioinformatics-powered process optimization. Global data is blunt. Strategic collaborations are the lifeblood of progress, enabling companies to pool resources, share expertise, and obliterate manufacturing bottlenecks that impede the advancement of lentiviral-based therapies. Isolation is a death sentence. And yet, for all the innovation and new partnerships, the central question remains unresolved. Is the current wave of technology and collaboration enough to break the bottleneck, or are we heading for a crisis of scale and safety as demand explodes? The next chapter in this race will be defined by bold bets, radical transparency, and a willingness to rewrite the rules of manufacturing. But as we look ahead, one haunting question stands out. What happens if the industry fails to deliver, leaving miracles stranded on the lab bench, just out of reach for waiting patients? In the second half of this story, we'll journey deep inside the labs and boardrooms where the blueprint for the future is being rewritten. Can the innovators win the race against time, cost, and complexity? Or will the hopes of a gene therapy revolution falter at the very final hurdle? Stay tuned, as the real battle for the future of medicine is about to unfold. As we look ahead, the stakes for the gene therapy revolution have never been higher. Innovation in lentiviral vector manufacturing is no longer just an aspiration. It's a necessity that will shape the future of medicine, the fate of companies, and the lives of patients around the world. If these latest collaborations, like Wacker Biotech and Expression Manufacturing, succeed, the consequences could be transformative. Imagine a world where highly personalized gene therapies are manufactured at scale, rapidly and cost-effectively. Patients diagnosed with genetic disorders, rare cancers, or immune deficiencies could receive curative treatments within weeks, not years. The idea of a new diagnosis no longer being a life sentence, but instead the beginning of a journey toward recovery, is finally within reach. But this path is fraught with risks and uncertainties. Even as Wacker and Expression integrate their best-in-class technologies, the industry as a whole faces a daunting climb. As Bioplan associates put it, upstream process development remains the Everest to be conquered for gene therapy manufacturers. Without efficient and scalable upstream processes, the entire field risks stagnation. The first scenario, a virtuous cycle of innovation is triggered. With robust, scalable technologies like the Plasmatector and lentiviral platforms, manufacturing titers soar, costs fall, and timelines shrink. This attracts new investment, fuels more R&D, and helps regulators gain confidence in the consistency and safety of viral vectors. Automated, single-use bioreactors become standard, analytical precision keeps risks in check, and stable producer cell lines finally unlock true mass production. Gene therapies move swiftly from trials to clinics, and the market, projected to surpass $5 billion by 2028, booms well beyond expectations. But what if the bottlenecks prove harder to break than anticipated? The second scenario is more sobering. Even with collaboration and new platforms, yield improvements stall below commercial viability. Regulatory pressures intensify after rare safety incidents, and without rigorous data and analytical controls, companies struggle to meet FDA requirements. Supply chain vulnerabilities, such as delays in high-purity plasmid DNA, trigger costly slowdowns. Investment dries up as costs mount and timelines drag. In this world, only a handful of the most well-funded players survive, while smaller innovators are forced to fold or merge. The promise of gene therapy remains largely unfulfilled for another decade. There's a third, and perhaps most likely scenario, a period of rapid progress punctuated by hard lessons. As McKinsey & Company warns, the ability to master viral vector gene therapy manufacturing will separate the victors from the vanquished in the coming years. Those who fail to innovate will be left behind. The companies that thrive will be those who not only embrace but drive radical, even uncomfortable change. They'll push for new approaches, insourcing plasmid DNA for control over quality and timing, automating upstream and downstream processes, and partnering deeply across traditional business boundaries. The broader consequences? Success here means more than financial returns or headlines. It could mean breaking the cost barrier that has kept gene therapy out of reach for millions. Think about it. CAR-T therapies have saved lives, but at $500,000 per dose, they're not scalable for entire populations. Affordable, reliable lentiviral vector production could usher in a new era where gene editing cures for sickle cell anemia, hemophilia, or previously untreatable cancers become routine standards of care, not medical miracles reserved for the few. Yet the ethical questions will only get sharper. As next-generation vectors enable precision targeting, even gene editing inside the body, society will need to confront questions about access, consent, and the boundaries of human intervention. The regulatory maze will grow more complex, not less, and each technical advance will demand new frameworks for safety monitoring, data transparency, and patient follow-up that stretch far into the future. So, what should you, as a viewer, take from this rapidly shifting landscape? Whether you're a patient, family member, investor, student, or simply a curious mind, the lessons of the lentiviral revolution matter now more than ever. First, understand that the technologies shaping tomorrow's medicine are being built today. When you read about a new clinical trial or a biotech partnership, remember, behind every news headline are teams of scientists, engineers, and regulators fighting to solve problems no one has faced before. Their success or failure will impact not only rare diseases but the entire future of healthcare. Second, realize that innovation is a team sport. The era when a single company or scientist could change everything is gone. Just as Wacker and Expression are pooling their expertise, the future will belong to those who collaborate across sectors, disciplines, and borders. If you're in the life sciences, whether as a student, professional, or investor, cultivating partnerships, staying open to new ideas, and embracing automation and analytical rigor will be essential to success. Third, keep a critical eye on hype versus reality. As Dark Horse Consulting cautions, a comprehensive and unflinching analytical strategy is the bedrock of product quality and safety in lentiviral vector manufacturing. Wishful thinking is not a substitute for rigorous data. For patients and families, this means asking hard questions about safety, follow-up, and access to new therapies. For investors and professionals, it means scrutinizing claims and demanding data, not just promises. For students, it means focusing not only on exciting new discoveries, but also on the painstaking work of process development, quality control, and regulatory compliance, the glue that holds life-saving therapies together. If you're considering a career or investment in biotech, the gene and cell therapy space offers one of the most dynamic, challenging, and potentially rewarding frontiers. But the blueprint for success is changing fast. Expertise in automation, in bioprocessing, in regulatory science, and in building bridges between disciplines will increasingly define who leads and who follows. For society at large, the rise of gene therapy underscores a deeper lesson, that progress is never automatic. Each breakthrough comes with new responsibilities, risks, and the need for vigilant oversight. We must balance hope with caution, speed with safety, and innovation with ethics at every step. So, returning to our core question, will the latest wave of technological and business collaborations, like the Wacker Biotech and Expression Manufacturing Partnership, be enough? The answer is both encouraging and realistic. Innovation is absolutely essential, but it will not be a magic bullet. The bottlenecks in lentiviral vector production are formidable, and only companies willing to experiment, automate, and partner deeply will push through to the next level. Some will break through and redefine what's possible. Others will falter, learning costly lessons that will, nonetheless, advance the field for all. The real secret is that progress will not be linear, nor evenly distributed. It will take a community of relentless problem solvers, courageous risk takers, and critical thinkers, inside and outside the lab. Here's a surprising fact you might not know. The very techniques now being honed to produce viral vectors for gene therapy are already sparking new waves of innovation in vaccines and in precision drug delivery. The advancements happening in this niche today could ripple out to transform how we fight everything from pandemics to antibiotic-resistant bacteria in the near future. The gene therapy revolution, powered by lentiviral vectors and driven by bold partnerships, represents the frontier where science, business, and humanity meet. The outcome is not inevitable. It will be shaped by the choices, investments, and discipline of this generation. So now it's your turn. What do you think? Will the innovators win the race, or are we heading into a bottleneck that could stall the gene therapy revolution? Let us know in the comments. Your insights drive this channel's conversation. If you found this deep dive valuable, don't forget to like, subscribe, and hit the bell for more expert breakdowns on the science.